This is my experimental setup for quantitative real-time PCR. As you see from this experimental setup, there is totally 48 wells, vertically 6 wells, 1, 2, 3, 4, 5, 6, and horizontally there are 8 wells, 1, 2, 3, 4, 5, 6, 7, 8. So according to this well, I just arranged my experimental setup as actin for resistance control samples. I do have 4 samples, as I told you in the previous video. So here RC, that means this is code for resistance control, resistance infected, susceptible control, susceptible infected. According to this, I just set up for resistance control actin, that means internal control gene, actin, that's 3 replications there. And for the second sample, the resistant infected or treated with pathogen, actin, there's 3 replications there. And the susceptible control, also the actin gene is replicated 3 times. For susceptible infected, that means susceptible infected inoculated with pathogen, actin is replicated 3 times. And here second gene, the gene that is differentially expressed in our sample according to the sequencing result. Defensing gene, the defensing gene is replicated for the RC samples 3 times replication. Defensing gene, and also for the resistance inoculated sample there is 3 replication here. Also for susceptible control there is 3 replication, and as well susceptible infected there is 3 replication. The second gene is pathogen resistance protein gene. This also replicated 3 wise, 1, 2, 3. For resistance inoculated, 1, 2, 3. For susceptible control, 1, 2, 3. For susceptible inoculated, 1, 2, 3. So replication is very important to reduce personal iraniasis. So we will take the average of the 3 replications, so it will reduce variation, it will reduce some personal errors. So for I should use here no template control. No template control for actin primers is also replicated 3 times. And no template control for defensing gene primers, replicated 3 times. And then no template control for pathogen related protein gene, also replicated 3 times. And the last wells are 3, that which is blank, without no reagent or without our samples. So here according to this, our experiment is set up. Here I just loaded my sample to 48 well reaction plates. This 48 well reaction plate is designed for this purpose. As I think the video is not clearly visible here, there is 6 wells vertically, and 8 wells horizontally. Totally 48 wells are there. So as my experimental setup, I just load my sample here, the RC sample, RI sample, SC sample, SI sample. Vertically, the 3 replication is for actin gene, and for control sample, for susceptible sample, for as well for control susceptible, for control infected susceptible. So according to this, I loaded all this reaction to 48 optical well plate reaction, or we can say that 48 well reaction plates. After loading all of my sample, that as I told you that here I do have total volume of 20 already loaded. So each well have 20 microliter of reaction, total volume of reaction, each well. Now after loading all of my sample, I need to seal with optical plate seal. This optical plate seal film, which is designed for this purpose and is used to seal this, the cover will be removed, the paper, the black, the white one is removed, so the plastic is transparent, that can easily pass the light through it. So it will be sealed like this. After sealing, we will take into the real-time PCR, quantitative real-time PCR. So this is a quantitative real-time PCR. So here I just put here, there is 48 wells here, 6 and 8. So my plates will be put here and then closed. So after this, I am going to set up my experiment on the software on the PC. In the beginning, there is setup, experimental setup. In the experimental setup, I need to name my experiment name. We call it defense and qRT gene. The experiment is set up. Which instrument are using to run experiment? Which instrument? So this is the biosystem real-time PCR. So it has 48 wells. So the 48 well option will be selected. Here the type of experiment we want to run. The analyze that the outcome should be, what the outcome should be. So what my objective here is to quantitate comparative CT value or delta CT. So I should select this option, but here so many options are there. Let me show you. As from the beginning, there is quantitation standard curve, there, melting curve, and quantitation relative standard curve, genotyping, and presence and absence of genes also there. So my objective here is to quantify gene. That means through the analysis of delta CT value. So this is what the experiment looks like. So here in the last, I am going to use the cyber green reagent. The cyber green reagent should be selected here. You can use also Tachman reagent, but in case of my experiment, I am using the cyber green reagents, and the reaction completed in 2 ways: 2 hours run and 40 minutes run. But I should select the 40 minutes fast run. So according to this, I just set up my experiment. Then the second one is target setup. My target setup is here. There is actin gene. Actin is my first target. Actin. Just I wrote here. Then I just added the facing. Fencing, and second target is qRT gene, as in pathogen related protein gene. Z gene. Here the facing gene, actin gene, I just set up the targets, write the target name here, and again I should add sample. I do have 4 samples here. RC sample, RI sample, SC sample, SI sample, and NTC, no template control for actin. No template control for defacing. And no template control for qRT. So here I just assigned my primer. I do have 4 primers, 4 samples: the resistant control, resistant infected, susceptible control, and susceptible infected. These are no templates for each primers. This no template for actin, no template for defensive primer, no template for qRT primers. So it should be saved here. Click save, and again click save. Now I just assigned my template, define my target and the sample. Now I should assign this. Here I have targets, 3 targets, and I do have the plates, so I should select 1, 2, 3, 1, 2, 3, 4. This is for select actin. Let's select it for actin, and 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. For PRB selected, and select for defensive. Facing selected. We select this, and this select, then select here. So the final one is the 3 plates. The final 3 plates is blank, no sample. Now I just assign my sample here. Sample polarity, second phase array, and SC here SI. This is RC, this is RI, this is SC, this is SI. Assigned, the sample is assigned. Now I should assign the no template for active. Is not the negative. This should be negative. You see here should be click negative. Negative. Totally I do have 36 unknown and 9 negative control. That's 36 samples with plus 9 negative controls. Totally 45 wells are now working. The 3-1 is blank wells. So here in the internal reference sample, I just RC samples selected, RC sample, and endogenous control 1 is actin gene selected. Actin. Finally, I am not using any relative quantification, any day going to be used here in my reaction. There is no passive reference day, so should be none. No passive reference day. The only day I am using this cyber green day that used to detect our gene quantified. So here I am going to use only cyber green day. I'm not using any passive reference day, so I just select none. Third one is running setup. Running setup, running natives. I just 94 for 2 minutes. For 2 minutes. For 2 minutes. This is the holding stage. Holding stage, the initial denaturation, 94 for 2 minutes, and the 40 cycles stage includes 3 steps, 94. For 50 seconds, 40 seconds, should be 94. 94 for 50 seconds, 60 for 1 minute, 1 minute, 1 minute, and after I should add for extension here. So 22. So 22 for 50 seconds, 15 seconds. And I'm not going to use the melting curve, so I should delete this setup. Delete it. Now I want to add holding cycle, after running for the purpose of gel electrophoresis. I should store at 4 degrees Celsius for 1 hour. For 1 hour I just store. So after this is very important for if I am going to run gel electrophoresis, I may store here at 4 degrees Celsius for 1 hour. Generally my setup is looks like this initial denaturation or holding stage we call it, and the 40 cycles. The first is 94 degrees Celsius for 15 seconds, denaturation, annealing of primers at 60 degrees Celsius for 1 minute, and the extension of the new template, new DNA strand from the old template at the temperature of 72 degrees Celsius for 15 seconds, and finally stored at 4 degrees Celsius for 1 hour. So this is 40 cycles. Okay. After running the setuping of running methods, I should go for reaction setup. My total reaction for this specific experiment is 20 microliter, and the jump spirit master mix concentration for jump cyber green mix is 2x, 2 times. The primers is to 20 nano picomole. So total reaction is as you observe 20 microliters. Okay, it looks like sample is diluted. So finally, after setup of reaction plates, material is there. Run. After completing the setup, experimental properties also are already set up like this, and the plate is also set up like this, and RAM is already set up like this, and reaction plate set up like this. After finishing this experimental setup, click here, run. Running the button, there is start run. So before I'm going to run this, I should save the reaction. Save as, and like this. I should save it in my folder, lab data, new folder, defensing, and RPG. So open. Save. Now saved. Then after saving this, my experiment name is defensing and PR revision, and then I click start. Wait for some minutes. There is a calibration, I guess. One or more calibration are expired. Do you want to continue? Yes. This loading to start. Now started. So now it's running. My experiment for real-time quantification is now running like this. At the end, in the next video, I will show you the amplification plots result here in the analyze part, the gene expression result here. There is relative quantification